Hi, welcome to A1.2 Nucleic Acids for IB Biology, new spec. This is the SL guide, so pause and read if you need to. Use it as a bit of a checklist in your revision if you need to as well. And then the HL biology guide as well. Remember that you need to not just watch the video, you need to do something with the information as well. Think about what works best for you and what's going to allow you to recall the information most efficiently. Okay, key terminology. The SL terms are green and HL terms are blue as normal. Screenshot this, make sure you are able to use all of this terminology confidently in your exam question answers. Okay, A1.2 nucleic acids. So we have nucleotides, which are the building blocks of nucleic acids, and they're made up of a pentose sugar here, a phosphate here, and a nitrogenous base here. Now your nitrogenous base can be adenine, guanine, thymine, or cytosine in DNA. And in RNA, thymine is replaced by uracil. Now you can see that thymine and uracil are very similar. So that's why uracil replaces thymine in RNA. And also just a bit of HL knowledge, because it fits nicely here, is that a purine always pairs with a pyrimidine. So adenine always pairs with thymine, and guanine always pairs with cytosine. The DNA molecule has a sugar phosphate backbone, which is made up of the sugar and the phosphate. So this section here makes up the sugar phosphate backbone. We don't include the base in that. So you can see here that the blue line is the sugar phosphate backbone. And you can see that here, this ladder looking molecule has been twisted into our DNA double helix. So the blue bits are still the sugar phosphate backbone, and the coloured bits in the middle are the base pairs. The nucleotides are held together by covalent phosphodiester bonds. So each nucleotide is held to the next one in the polynucleotide chain by a phosphodiester bond. And the sugar phosphate backbone helps to give the molecule strength and prevents the bases from being exposed. RNA is a single-stranded molecule and it contains uracil, like we previously said. The pentose sugar in RNA is ribose, so in DNA it's deoxyribose, in RNA it's ribose. RNA is also a polymer and it's made up of repeating nucleotide monomers, so the RNA nucleotide looks very similar to this, but this sugar is a ribose. In DNA, this sugar is a deoxyribose. The bases in DNA, as we said, are adenine, guanine, thymine, and cytosine, whereas in RNA, adenine, guanine, cytosine, and uracil. Now we need to be able to compare DNA and RNA. So DNA will contain thymine and RNA contains uracil. DNA is a double-stranded molecule, so you can see here these are double-stranded molecules. We've got one polynucleotide chain here and another here. Your bases are joined by hydrogen bonds. There's 2 hydrogen bonds between A and T and 3 between C and G. RNA is a single-stranded molecule. DNA has a deoxyribose sugar, as we previously talked about, and RNA has a ribose sugar. Okay, HL DNA structure. So in one strand of DNA, there is a 3' end and a 5' end. So you can see on this diagram here that we have a 5' end here and a 3' end here. Now, all this means is it's the carbon within the sugar that the phosphate that is sticking up here is attached to. And down here, it's the 3' carbon that is in the corner here. The 5' carbon is in the corner here. And you can see that this strand has a 5' end here and goes down to the 3' end at the bottom. And that means that on this side, the 5' end is at the bottom and it's going up to the 3' end at the top there. So we say that DNA is an anti-parallel molecule. So it's anti-parallel because 5' end here, 3' end here. And then opposite that is a 5' end going up here to the 3' end, which is opposite this one. You can see as well that these sugars, these ones, the oxygen is pointing kind of upwards, and on these ones, they're flipped and the oxygen is pointing downwards. DNA is also wound around cores of 8 histone proteins. And when you wind the DNA around the histone proteins, we call that nucleosomes. and nucleosomes allow the large DNA molecule to fit into the nucleus. Without those histone proteins and the ability to wind the DNA around them, the DNA wouldn't be able to fit into the nucleus, and that would obviously be an issue. Okay, finally, we've got, not finally, we've got Chargaff's rule. And Chargaff thought that DNA was a tetranuclear. It was thought before Chargaff that DNA was a tetranucleotide molecule, and that means it's repeating sets of 4 nucleotides. Now, Chargaff thought that this probably wasn't true, and so he investigated the number of each different base in different DNA molecules using paper chromatography. Now, what he found, you can see it in this data here, is that there were very similar numbers of A and T in each organism, and there were very similar numbers of C and G in each organism. So although they're not exact, they are very similar. You can see same for the human, fruit fly it works, wheat it works, fungus, rat, E. coli, and so on. Now, because the bases did not exist in equal quantities and they did exist in similar numbers for A and T and G and C, that proved that DNA was a highly variable molecule, and it also proved the DNA base pairing theory. Therefore, A would always pair with T, C would always pair with G. That meant that it was a double-stranded molecule, and that sequence was different for every gene. Okay, finally now, we've got the Hershey-Chase experiment. Now, Hershey and Chase were trying to prove whether DNA or protein were the hereditary material that organisms passed from one generation to another. Now, they did manage to prove that DNA was the genetic material that's passed from one generation to the next, and we still accept that theory now. Now, what they did was they used a bacteriophage, which is a virus that can infect bacteria, and they used E. coli. So obviously this bacteriophage is able to infect E. coli. The bacteriophage has a sulfur-containing protein coat and phosphate-containing DNA. So DNA always contains phosphate, as we saw in the SL content, but it doesn't contain sulfur. And again, protein contains sulfur, but it doesn't contain phosphate, and that's really important in this investigation. In the first investigation, the bacteriophages, the viruses, were grown in a culture that had a radioactive form of phosphorus, that's phosphorus 32, and as those viruses replicated, they would incorporate phosphorus 32 into their genetic material. That was then incorporated into the bacteriophage, and then when the bacteriophage was mixed with the E. coli, the bacteriophage would infect the E. coli by injecting its genetic material into the E. coli. And then, as we know about viral replication, the bacteriophage's genetic material is incorporated into the E. coli, and then the E. coli makes copies of that. In the second investigation, the bacteriophages were grown in a culture containing a radioactive form of sulfur, which was incorporated into the protein coat of the bacteriophage. Now, when the bacteriophage was mixed with the E. coli, that protein coat was not passed on to any subsequent generations of the virus. So what that told us was that because the phosphorus was present in the next generations of the E. coli after the bacteriophage had infected it, that meant that the DNA was the genetic material that was passed on from one generation to the next. The protein wasn't then present in subsequent generations of the E. coli, so therefore Hershey and Chase could deduce that the protein was not the hereditary material. There's a little graphic here if you need a graphic.